started RAIN as a community DIY incubator, and our goal really was focused on training people. My background personally is in infectious disease. I've been working in infectious disease for more than 30 years. I've been involved with the government, the Army and DoD, and pandemic preparedness, and have really, my effort has been on training. Coming back to RAIN Incubator, again, continuing my funding with the military, I was allowed to kind of set up my own nonprofit. And surprisingly, this has been very operational during this outbreak. We were able to stay open when many other universities and hospital labs and even government labs had to close down. And most viral diseases, emerging diseases, come from somewhere. That's the ones that plague human and agriculture. So 70%, that's really a number that we've really kind of seen in these surveys. And so when a virus appears, when a new pathogen appears, it has come from somewhere. And because of what the work we've done in terms of being able to sequence and classify all the viruses in our groups, we've been able to rapidly identify where these pathogens come from. So this new SARS, the novel SARS, SARS-2, is no exception. It's believed, the consensus has been, it emerged in China, likely in a wet market. It likely, like other coronas, the 7 coronaviruses that circulate in humans, emerged in bats, and it is very closely related to the originals, the SARS-1 we had worked on in the early 2000s. And my mentor, Ian Lipkin, we actually went to China. We went there for that response and studied it. So we had a lot of experience with coronavirus, and we know that these coronaviruses are, in addition to being respiratory diseases, are also spread through fecal routes. And so a lot of the receptors that are in the lungs are also present in the gut. And so it was just like SARS-1, it was logical to assume this would also be in feces. When I was working with Ian Lipkin at Columbia, this is a lab that has a rich history of really discovering viruses, actually other pathogens, but we've really looked in all animals and characterized them because we know that if they're going to emerge in humans, they're going to come from somewhere else. So this is just an example. This slide is an example of all the viruses that have been discovered and characterized at Columbia University. But another approach that we're very interested in going after is really a serology approach. So the molecular tells you where the virus is, or if somebody is infectious, or where it is in the environment, if you're looking out in the environment. The serology tells you who has seen the virus, and it gives you almost a memory of anybody who's come in contact with it and fought off the infection. You will have kind of a classic antibody response. We've actually synthesized the peptides that could recognize the serology. The main ones we know from previous serology and antibody arrays that we've built is really some of the basic proteins. We've made M protein, S protein, E protein. And so you can look at either the full protein or small peptides that are subunits of that. And so in the S proteins, there's usually the S1 and S2. We found that the S1 is the best. That includes the spike, and it also has these ACE2 receptors, which is part of that. And then also a really good protein is the nucleocapsid. So making peptides that cover regions that are unique to the SARS-2 virus that are important. Just reviewing antibody response to COVID infection, this is kind of your classic immune response. We usually, upon a virus being expressed in the body, people have symptoms, but many people can be symptom-free. We all will make a very rapid IgM response to the virus, and IgM tends to be very sticky. It's also not effective, but it is a good first stopgap. What happens then after the course of 6 or 7 days is you start seeing an increase in IgG antibody as many of the antibodies go through a class switch. antibodies tend to be higher affinity, at least independently, and they also are actually much more effective in neutralizing a viral infection. So these will peak at 12 to 13, 14 to 15 days in individuals. And then as memory cells are made, these are the antibodies that persist, and you'll have always circulating levels of IgG. And so if you interrogate somebody who's gone through an infection, you'll see high levels of this, making a design for individual peptide arrays. So you can create a slide. Currently, the current slide is 6 million spots can be printed down. And it's basically a 20-color printer, and I can make 16 mers of peptides. And so the strategy here is to do PFAM alignments of all human coronaviruses, and we will probably include one bat one that has occasionally moved into humans but never persisted. But we'll walk along all these sequences and print down every possible combination that we would see. So all these antibody epitopes essentially go down. And this gives us, when you screen serum across it, will give you a very fine output of all the epitopes that are responding that would be specific for, in this case, SARS-CoV-2. And so that's going to give us a better determination. Once we have that, then we can report out those sequences. And when we move, our strategy really is to move away from ELISA-based assays we want to go more to something that would move into a CLIA lab like the Ella or JEST platforms. And so we can report out. And so the JEST would be good for one application. We're looking at like an addressable array, but Ella looks very promising for being able to print down all the peptides that would give you a very specific antibody response to SARS-CoV-2. See, this is the assay characterization with JEST. I have some familiarity with working with lysates on this. And so I think the JEST is promising for looking at specifically lung lysates or intestinal lysates. For further on, when we're looking at maybe more defined antibody responses, we're trying to look for neutralizing antibodies. I think this is going to be a very promising platform for that. Also, because we can work in kind of a semi-fluid phase, it's going to be great to work with some peptides that may be very difficult to keep in, like to adhere to solid surfaces. So this is one of the reasons we are excited about the JEST. And then with Ella, this is what we're looking for as the possible way to move from a peptide array or an ELISA onto a surface that we think is going to be good for putting into a clinical lab. And so again, picking the right antigens that we can spot down, we can likely be able to show a really high sensitivity and specificity for certain peptides. And we'll also be able to tease out even IgM and IgG antibodies from the host. So this is our goal is really to start with open cartridge and then move to the closed cartridge system so that this could be mass produced, like on a really high quality at a company level.
So right now we are preparing to both develop assays to detect the virus itself, which we already have implemented, and now we're optimizing, and then we're also developing assays to be able to detect previous infections, looking for the antibody specific to the SARS-CoV-2 virus.
So our goal is really to develop detection assays or alternate ways to do that. Serology, which is another pillar that we're going to go after to look at the protection or circulating antibody in the population. So de-identified samples we could work with and then basically give that information back to the authorities to understand where this virus is and who's protected from it or who's seen it.
These are the people that have really played a huge role in this. So so really our RAIN members from our lab, Dan Langevin, Jenna, Ryan, my two Ryans, and in my group. And then I really want to credit, Biotechne, the scientists on board, Annegret and Phil, who's supported our effort, Kelly for loaning us her scanner, and Chris and Francisco. I really appreciate it.